Okay, so we've looked at a lot of different flies. Now we're going to end our discussion on the Diptera by looking at flies that are a little on the edge of forensic importance, but they might be becoming more important, and at the very least, they are super, super annoying. We are looking at the mosquitoes, or the Culicidae, or Culicity, depending on who you talk to. So, the name Culicidae is derived from the Latin culex for gnat. And this is the type specimen for Nematocera. Remember Nematocera way back in the day? So this is the type specimen for that group of flies that have those long antennae, those sort of elongate bodies, usually proboscis, that sort of thing. So the Culicidae are the mosquitoes, and they consist of about 3,200 species of mosquito out there, but we really know that there are more to be discovered, especially in the tropical rainforest. Just like we saw with the 40 and with most of these insects, those tropical regions, they're much more diverse. And so we're going to find new ones down there. They're also much less well surveyed than in temperate regions. This is because in temperate regions, they seem to come into contact more commonly with humans, and therefore we care more about them. Now, surveyed species often reveal that they consist of something that we call complexes. This is an interesting concept in entomology, a species complex. So remember back to the very beginning when we talked about what is a species, what actually makes a species. That is still up for debate because of course it is. With these ideas of species, and when we're talking about some of these flies that they are so in-depth researched, I guess is the word I'm looking for. They're researched in such depth, we're finding that we're actually watching evolution happen. So a species complex is a group of very, very closely related species. They're so incredibly close, in fact, that we cannot tell them apart morphologically. They may be reproductively isolated, so we only see them mating with one another that live in one particular area. They might be regionally isolated, so there are certain species complexes where you have these two species, one that likes it cold, one that likes it warm. So you don't really see them being able to mate with each other. But if you were to put them in the lab and look at them under the microscope, they look identical. And then they will mate with each other and produce viable offspring. So they do meet our definition of the biological species concept, but they just never mate with each other in the wild. Or maybe they're niche-specific forms. So maybe there's a particular type that only feeds on monkeys, or only feeds on humans, or only feeds on birds. So they never run into the other members of their species complex. So because of this complexity, because of this ridiculous way that we're actually watching these mosquitoes evolve literally in front of our eyes, we have to come up with a new way to talk about them. And that's when we come up with the species complex. So it is a group of really closely related species, so closely related that if we put them in the lab, they're still able to mate and produce viable offspring. They are morphologically identical, but in the wild, they do not interact for some reason. So we're actually watching them speciate. So we talk about species complexes a lot just to indicate that there is some sort of isolation among the groups within this particular species. So if you read about that or if you hear me talk about it, that's what I mean. All right. So basic morphology of the Culicidae, of the mosquitoes. The eggs of most mosquitoes are elongate, and they're either ovoid or spindle shaped. So they're pretty straightforward. They look like eggs. Now the outermost egg or shell of the egg is usually has some sort of intricate surface patterning. So these are what the eggs look like right here. And if you look at them underneath the microscope, they will have these crazy patterns on that chorion or on that outside, that shell of the egg. And these patterns can actually help you diagnose a species or tell the species from the egg stage, which is amazing. It is really hard to tell species from eggs. Most insects, but mosquitoes, it is possible. So those of you, if you start getting interested in medical entomology, you will go into mosquitoes That's pretty much where it's all at right now. And you can tell your species just by looking at the eggs under their microscope. So that's pretty neat. Now, some species eggs have these air-filled compartments that serve as egg floats. And this will allow the eggs to sit on the surface of water and just sort of float around singly. Others are oviposited together in these big clutches, and they're glued together and create a little raft. So they are primarily laid in liquid. There are a few that will be laid on solid substrates that are going to flood at some point. Now, the eggs will hatch into the larvae, and the larvae are known as wigglers. So that's because if you've ever seen mosquito larvae, that's what we're looking at here. They just sort of wiggle around in there. So people call them wigglers, because we're good at things. And they pass through 4 larval instars. And so unlike the Culicidae or a lot of these other brachyceran flies that we're looking at that have 3 larval instars, the mosquitoes have 4 larval instars. Now, they closely resemble one another. When we're telling the larval instars apart, the major difference is their size. So the first larval instar is very, very tiny, and they just get bigger and bigger. Now, for individual species, it is possible to tell the larval instars apart with morphological characteristics, but there's nothing like the 1, 2, or 3 spherocular slits that we see in Culicidae. So it's just mainly size that allows us to tell them apart. Now, the mosquito pupae are known as tumblers. So this is because they tend to float at the surface of the pool or the water where they're in, and then they tumble down to the bottom when they're disturbed. In general, the tumblers or the pupae are comma-shaped. So the head and the thorax fuse to form a cephalothorax. Remember the word cephalothorax from way back when we did classes? Yeah. So the cephalothorax, and then the abdomen curls beneath it. So these little darker dots in this picture, those are actually the pupae. See how they're kind of comma-shaped while the wigglers are more just elongate? Yeah, these comma-shaped things, those are the pupae. Okay, so that head and thorax are in the cephalothorax, and then they have this pair of respiratory tubes or air trumpets that project from their dorsal surface that allows them from the mesothoracic region, that allows them to get oxygen at the water surface. So they just sort of sit at the water surface, they have these trumpets on their mesothorax, and they get air. Now, the pupae can flex its abdominal segment, so it is a mobile pupae, unlike the Culicidae and the Muscidae and these others that we've seen. So if they are in danger or they're threatened in some way, they'll flex those abdominal segments, and they have these 2 little abdominal paddles at the on the last segment of their abdomen, and those will sort of flap downward, and it'll propel them backwards through the water, so they're able to move around. That's where they get that name tumbler. They tumble through the water, and they're able to get away from danger. Now, the adults will emerge from those pupae at the surface of the water, and the adults are slender. They have very thin legs and very narrow and elongate wings. Their body surface tends to be covered with scales, with setae, with fine pile, and a lot of these scales and setae and things create characteristic markings and colors of each one of these species that'll help us identify these species underneath a microscope. Normally, they have these 2 compound eyes, which will wrap around the front and the sides of their head, and they have these antennae that arise between the eyes. Their antennae tend to be long and filamentous. Now, the antennae are usually sexually dimorphic, so this is what allows us to tell the male from the female mosquito. That's what sexual dimorphism means. So in the males, in most species, the antennae are plumose. So you can see that right here. So they have all these extra hairs coming off of them. This is because the antennae the males will use their antennae to find female mosquitoes, to cue in on the pheromones of the females. While the female mosquitoes have the filamentous antennae with maybe some sparse whorls of hairs around those antennae. But when you look at them overall, big old plumose antennae, that's a male mosquito. Long, slender antennae, that's a female mosquito. Neat. Now, the mosquito proboscis is very prominent. So the mouth parts are elongate into this proboscis. They project anteriorly, and they are at least 2/3 the length of the abdomen. So they're very, very long proboscis. Now, the female have appendages in their proboscis that allow them to pierce skin. So they're able to get blood. So the females feed on blood. The males do not. So the males instead use their proboscis to feed on nectar. Now, the mouth is fitted with these very large salivary glands that aid in digestion of food. In the females, it's extremely complex. If you were to take, say, my medical entomology course, we'd actually go over all the different lobes of the salivary glands of the mosquito in the female and talk about the type of digestive enzymes that are found there. There are some that are there to digest sugar, others are there to digest proteins and things of that nature. The males have a much more simplistic salivary gland setup because they only feed on nectar. So they only need sugar digestion in their saliva. So you see this differences in the mosquito salivary glands as well as in their proboscis and in their antennae. Now, the abdomen is clearly segmented on both the males and the females, and it's capable of extensive expansion along the pleuron, along the sides. So there's a membranous area in between each set of tergites and sternites. Again, remember what those are. The tergites are those plates on the dorsal surface, and the sternites are the plates on the ventral surface. So it allows for expansion of the abdominal wall to accommodate large blood meals, large sugar meals, and allow the females to develop large clutches of eggs. So their abdomens can expand exponentially. So they are holometabolous insects. They go egg, larvae, pupae, and adult. Now, the stages of life are completed in two different environments. The larvae are aquatic organisms, while the adults are terrestrial organisms. So it's a little bit like those hemimetabolous metamorphosis, except this is holometabolous because they have a pupal stage. So the larvae and the pupae will develop in a wide range of aquatic habitats. We'll see them in tide pools, in rain pools, in flood water, in streams, swamps, lakes, in tree holes, in fruit husks, mollusk shells, in drinking water pots, in discarded tires, really anywhere where there is a little bit of water that is there long enough for the larvae to develop, that's where these mosquitoes will develop. So the only requirement of all the developmental sites is they maintain at least a film of water for the duration of the larval and the pupal periods. So they don't need a lot of water. And this is why mosquitoes are so good and so freaking annoying, especially in areas where we get a lot of rain. Anytime we've got a rain event, it's going to fill up these temporary containers, and anything as small as even a snail shell can hold mosquito larvae. So there are individual species, though, that will oviposit in characteristic developmental sites. So these sites that have characteristic physical and chemical properties. So there are some that like certain areas. They like certain amounts of organic material in the water. They like really polluted material, water, really clean water, whatever. So it just depends on the species. But overall, they just need a film of water to complete their life cycle. Now, eggs are laid either in the water or on solid substrates that are likely to become flooded. So the eggs begin as white, but then they turn dark within hours as the eggshell tans. Now, the eggs usually complete development within 2 to 3 days after being oviposited on water. They may take a week or so in cooler climates. So if they are on water, they can go pretty quickly in the warm areas, 2 to 3 days, but up north or in the cooler months of the year, it can be a few weeks. Now, the eggs that are laid on solid substrates out of water that are there just waiting for a flooding event, they will remain in the egg stage until that water is present, until they are flooded. So that hatching happens relatively quickly after a flooding event. So you can get these mosquitoes that will lay their eggs on the sides of containers or on old tires or something. And as soon as it rains, the oxygen level in the environment is going to lower. That'll alert the eggs that, hey, there's water, and then they will hatch very, very quickly after that happens. That's why after a rain event, you tend to see mosquitoes really fast because they're just waiting and ready to go. Now, after hatch, the larvae are going to spend most of their time either at the water surface or at the bottom of the water column, and they'll just come up for air occasionally. Under ideal conditions, the larval phase lasts about 5 to 6 days. So male mosquitoes take a slightly shorter amount of time. They will pupate about 1 day earlier than the females, so a lot like the phorids. In non-ideal conditions, the growth rate is much slower. So if it's nice and cold or something happens. And so depending on how bad the conditions are, then the larvae can take weeks to months to develop. And there are some larvae that can tolerate being frozen in ice for short periods. So we'll see these larvae in these ponds. If it ices over, they can tolerate that. And as soon as it thaws, they'll continue developing. So that's why, again, we see mosquitoes so quickly after a thaw. Now, the pupae spend nearly all their time at the water surface. The pupae are very dark. They're pretty sclerotized, so that's why you see that sort of dark comma shape in the pupae. And in warm water, the entire pupal stage can last about 2 days, so it doesn't take very long. In lower temperatures, it's much longer, of course. So again, the adult males emerge earlier than the females, and as emergence approaches, the pupae are going to remain stationary at the water surface. The abdomen is going to gradually straighten out over 10 to 15 minutes, and the adult is going to emerge from the pupal cuticle by ingesting air, causing the cephalothorax to split along the dorsal surface. So there's a weakened margin along this dorsal surface. They'll pop open that dorsal cephalothorax, and then they're going to push themselves out of the cuticle and stand on the surface of the water. Now, the newly emerged adult is capable of only a very short flight until it becomes fully sclerotized. So it's going to fly away out of danger, so there's no fish or anything that's going to be eating it, and then it's going to sclerotize. Then those larval energy reserves provide sufficient energy for a few days of flight and survival. So within those few days, that adult then has to find sugar and protein in order to continue its life cycle. Now, the first 3 to 5 days of life in the adults, they need to find sugar. And they will usually feed on nectar or honeydew, so that they will use these carbs to become sexually mature, and then they're going to mate. The sugar feeding is repeated throughout the adult life cycle for both the males and the females. Now, females typically mate only once, but the males can inseminate several females before their supplies of mature sperm is depleted. Only the females are going to feed on vertebrate blood, and they need living blood in order to feed. In most mosquitoes, ingestion and digestion of the blood meal initiates egg development, so they are anautogynous. So they need a blood meal in order to produce eggs. So they feed on this blood, and that's going to stimulate this cascade of hormones from the brain and the ovaries that is going to stimulate egg production. But they only produce eggs if their blood meal is sufficiently large and if the female's ovarian follicles are receptive. So she may not be able to develop any eggs until she's had, say, a sugar meal or she's had a preliminary blood meal. It just depends on the species. Now, once everything is good to go, she's going to produce a single mature clutch of eggs after each blood meal. So a blood meal is required for each egg laying cycle. Now, all the eggs in her ovaries are going to develop all at the same time, and they're going to mature about 2 to 5 days after a good blood meal. After that, she's going to go find a place to oviposit. The number of eggs that she can oviposit depends on the size of the female body, the energy reserves, and the size and quality of that blood meal she was able to take. Now, eggs are laid on either dried substances that are flooding or on the surface of water. If they are laid on these dry substances awaiting a flood, they may only hatch a portion of a clutch at a time during any one flooding event. This is called installment hatching. So it's the combined result of intrinsic variation among the eggs and their hatching stimulus threshold. So it's basically the female isn't putting all her eggs in one basket at all. She is laying all of her eggs in one place, but maybe that first flooding event only causes a few of them to hatch. It takes a couple of more flooding events for the rest of the clutch to hatch. So you're not going to see them all hatching all at once. You're going to have this installment hatching. Now, during hatching, the first instar larvae uses a dorsal hatching spine on its head called the egg breaker or the egg burster. It's going to apply pressure to a preformed weakness in the eggshell, and this is going to cause the eggshell to pop open at one end, and that larva is going to wiggle free. Now, the larvae are going to feed on a variety of organic detritus, on suspended material, and maybe small organisms in their aquatic habitats. These include things like bacteria, like protista, like fungi, algae, invertebrates, small macroinvertebrates, all those sorts of things. Usually the detritus is dead plant material, maybe little tiny invertebrates, maybe bacteria, that sort of thing. And they're going to collect these items by filtering, by gathering, by scraping, shredding, and swimming after them and preying upon these organisms. Now, the pupae usually remain motionless at the water surface or at the base, and they just sort of set the tips of their thoracic air trumpets in contact with the air. If they're disturbed, and they're going to be disturbed by movement, they're going to be disturbed by shadows, so a quick change of light, that's going to bother them, and they're going to move to the base of the water column. Each species has a characteristic pattern of daylight activity. Usually, the adults will have 1 or 2 flight periods that occur each 24 hours. And the adults are characterized as being diurnal, so during the day, nocturnal, so during the night, or they only fly at dawn and dusk. So during these periods, both the sexes are going to take flight. Both the males and the females are going to go out flying. They're going to search for mates. They're going to search for food. Now, the dispersal of some mosquitoes is only a few dozen meters from their larval habitats, and at most, they're going to fly less than 2 kilometers in their lifetime. So that is some of the mosquitoes. Other mosquitoes will enter a specific dispersal mode. It's usually wind-assisted, and it'll carry them dozens or even hundreds of kilometers from their origins. So those that are able to invade new areas are usually really good at dispersing like this. Others will just stay in very small pre-described areas. Now, mating usually takes place a few days after adult emergence. The males will typically form these little flight swarms at particular times and places. So they will find either really obvious environmental cues like rock faces or big mountains or things like that, or less obvious places. We've seen mosquito swarms happen above certain stalks of corn, or sometimes above their potential hosts for the female. So if you are really, really attractive to mosquitoes, you will get mosquitoes just flying over your head waiting for mates. So you are the swarming area. Awesome. So what happens there is these males will find these flight swarms, and each male is going to follow this looping flight path. A female is going to enter the swarm, and then the males are going to detect the characteristic frequency of her wing beat and her wing position with their plumose antennae. So they cue in on her wing beat, and that is characteristic of a particular species. So people have actually developed apps for your phone that will record the wing beats of a mosquito and determine the species based on that frequency. That's amazing. So then they're going to cue in on their conspecific or their same species female. They're going to pursue her for mating. And if the female of a different species entered the swarm, the males either do not respond to her, or they're going to release her immediately after mating. So they're just not going to mate with a different species. Now, copulation lasts from 12 seconds to several minutes. Again, so many jokes I can make there, but I won't. Then the male's going to release the female. She's going to go off and try to oviposit or find a blood meal. Now, both males and females regularly feed on plant sugar throughout their life, but only females feed on vertebrate blood. So water is presumably taken from the surface of moist substrates, as well as in sugar meals and in blood meals. So they feed on liquid primarily, and that's how they get enough water. Mosquitoes are going to hunt for food and host through both sight and olfaction. So just like we saw with the Caliphoridae. They begin their foraging flights in relation to specific visual features along the horizon, or they begin flying crosswind and downwind, trying to pick up on their olfaction, where they're going to find these likely sources of sugar or blood. So when they see these likely sources of sugar or blood, they're going to alter their flight paths and move directly towards the objects. If they detect the odor of flowers or the odor of hosts in the absence of visual information, they're going to turn and fly upwind within this downwind drifting odor plume, eventually arriving at the odor source. Female mosquitoes rarely begin responding to vertebrate hosts and taking blood meals until 1 to 3 days after adult emergence, and not until after mating and sugar feeding. So they're not ready to bite you right away. The hosts of these females, they include all classes of vertebrates, from mammals to birds to reptiles, amphibians, fish, whatever. And host specificity and preference varies depending on the species. So there are some species that feed almost entirely on members of one genus of Animalia, while others will opportunistically attack any vertebrate, any blood holder that they come into contact with. Now, one of the ways that we can use mosquitoes in a forensic capacity, one of the ways that people are looking into this, is you can actually analyze the blood meal in the abdomen of the female. So here is a recently fed female. You can see that she's got blood in her abdomen. If we can get her early enough, before all of her digestive enzymes break down that blood meal, you can analyze what animal this came from. There is some definite potential to get an individual person from this. So a lot of work has gone into that. This is sort of the next level forensic entomology type of stuff, is analyzing the blood that these mosquitoes have imbibed to try to figure out what it is they're feeding upon. Okay, so that is one of the ways that we can use these things. So host finding behavior in mosquitoes involves the use of volatile chemicals to locate hosts. They're going to cue in on things like carbon dioxide and lactic acid. They're some of the best documented host attractants. There are other skin-associated odors that are known to be important. We think that they're associated with the bacteria on the skin, and these odors have an effective range of 7 to about 30 meters. In some species, they can pick them up 60 meters away. But the reason this is important is because there are some people that are super, super attractive to mosquitoes, right? You might be one of those people. You're sitting outside and there's mosquitoes about, they're going to feed on you. Other people just are not bitten by mosquitoes. So my husband is very attractive to mosquitoes for some reason. I am not. I say it's because I'm an entomologist and I have an understanding with them, but it's because we have different bacteria on our skin. So as we're sitting outside, there can be one mosquito within, 60 meters of us, and they're going to find my husband. I don't even notice that there's mosquitoes out there. So we can be sitting outside, enjoying the evening, whatever. And Dina will go, we have to go inside. There's mosquitoes. I'm like, oh, are there mosquitoes out? You look at him, he's covered in mosquitoes. There's not a single one on me. So, yeah. So this is the other reason I want to bring this up, is when you are out at a crime scene, mosquitoes are going to be a problem. Just the end. You need to know if you are super attractive or not to mosquitoes, and you need to be careful about their biting, because they do carry disease. They do vector a lot of nasty diseases. And there are quite a few here in the US that can cause problems. So if you are outside at a scene, one of your normal precautions you need to take is to protect yourself from mosquitoes. So mosquito repellents work by blocking these host-associated odors that the females pick up on. So you're trying to block that CO2, that lactic acid, those odors from the bacteria. Those of us who aren't very attracted to mosquitoes, we can get away with those really nice smelling mosquito repellents, or those really natural ones, lemongrass and Skin So Soft from Avon and all those sorts of things. People who are super attractive to mosquitoes, you try one of those things and mosquitoes can be like, okay, nope. And they're going to be able to smell you through it. So you have to get the really hardcore stuff, the really hardcore DEET that you can get at Academy or at REI or some of those other outdoor stores. And you usually want something with about 40% DEET in it. If you go to a tropical area where it's really, really bad mosquitoes with really nasty diseases, you want to get 100% DEET. And 100% DEET is crazy. It'll take your nail polish off. It'll melt paint. It's nuts. But you need to wear it so you don't, die of yellow fever or something of that nature. All right, so that's the basics of mosquitoes. That's really, I just wanted you to know how they worked, where they lived, because you will run into them when you're out and about. And sometime you will get called in on a case that will have mosquitoes there. So I just wanted to touch on that a little bit. Let me know if you have any questions.